So we now welcome the medical laureates to the Faisal University's Pioneers in Science Day. We are honored to have Professor Dame Sarah Gilbert, the co-laureate of the 2023 King Faisal Prize in Medicine, presenting her lecture titled, Preparing for Pandemic Vaccine Development. Professor Dame Sarah Gilbert is the Said Chair of Vaccinology and Head of Outbreak Pathogen Vaccines Group in the Pandemic Sciences Institute at the University of Oxford in the United Kingdom. Her chief research interest is the development of viral vectored vaccines that work by inducing strong and protective T and B cell responses. She leads work on influenza vaccine development as well as vaccines for many different emerging pathogens, including Nipah virus, MERS, and Lassa virus. She co-developed the Oxford AstraZeneca COVID vaccine in a very short period of only 10 months. This vaccine is a low-cost, easy to ship, and has been administered to over 2 billion recipients worldwide. Please join me in welcoming Professor Dame Sarah Gilbert.
I also have a Saudi student in my lab currently, Dr. Sarah Batawi, and prior to that, another student, Dr. Naif Al-Harbi, also worked with me in Oxford, and while he was studying and working at Kaimark, and we worked together on the MERS vaccine development, which is part of this story that I'm going to tell you now. So what we're going to go through is a bit of background as to how I got into developing vaccines against emerging pathogens, how this became really important in 2020, and what I think the future is now for my lab in particular as we move into the post-pandemic period. So I want to take you back to 2014. Now, at the very end of 2013, an outbreak of Ebola started in West Africa. It wasn't really noticed very much by most people in the world until the middle of 2014. And this was by no means the first time that Ebola virus had caused an outbreak in either West or Central Africa. It was first discovered in 1976. There are two main forms of it, Ebola virus Zaire and the Sudan form also, and one or other of these had caused an outbreak, usually small in isolated rural areas, but this had happened roughly every year since 1976 when this virus was first identified. And those small outbreaks were dealt with by identifying the contacts of the patients who were infected with Ebola virus and quarantining them until transmission between people had ceased. But in 2014, that didn't work, and the virus managed to get into populations in cities where there is much more opportunity for rapid person-to-person transfer of infectious diseases. It spread across borders into 3 different countries. It was starting to spread into the rest of the world with sporadic cases occurring in people who traveled to the US or to Europe. And finally, it was decided that some very different approach needed to be taken to control this outbreak. The number of cases were increasing exponentially by the middle of 2014. And it's not that there had been no work to develop a vaccine against Ebola virus, but the vaccine development work that had been done was really done with the aim of producing a vaccine to use to protect people in the West in the event of a bioterrorist attack with Ebola virus. Vaccine development had moved slowly. These vaccines had never been tested in the area where outbreaks of Ebola virus actually occur. But there were vaccines that had been brought through the early stages of development. In particular, one based on a simian adenovirus had been manufactured and was almost ready to go into clinical testing. The difficulty with accelerating this clinical testing was that the adenoviral vector, the safe version of the adenovirus originally isolated from a chimpanzee that had been used to make the Ebola vaccine, was of a type that had only been tested once in humans previously. It was called ChAd3, for chimp adenovirus serotype 3, and a ChAd3 vectored vaccine against hepatitis C vaccine had been tested in one clinical trial in the clinical trials unit that we work in Oxford. And so it was felt that the fastest way to get the ChAd3 Ebola vaccine into clinical testing would be to work with our group in Oxford, with the ethical committee, with the regulatory authority that had seen this use of this vectored vaccine before, with a group who knew how to complete this trial. And so although it wasn't a vaccine that we've been developing from scratch ourselves in Oxford, we were asked to join in with this effort, and we very rapidly agreed, and with funding from the Wellcome Trust, we managed to arrange to have the vaccine put into vials, the final stage of vaccine manufacturing that's needed before you can start clinical testing. We did the very rapid submissions to the regulatory and ethical authorities. They prioritized it. They had special meetings just to review the dossiers. They approved quickly. We got the vaccine labels put on the vials, signed the contracts, and by the middle of September, so only 1 month after being asked to become involved in this initiative, we started to vaccinate people in Oxford. And a month later, the 60 people planned for this clinical development had been vaccinated. This meant that we had data on the adverse events after vaccination, whether people had a headache or a fever or a sore arm after vaccination, and we had data on the immune responses in the people who'd been vaccinated with a single dose of this vaccine, showing that they had a good level of antibodies against the Ebola virus. Now, we can tell that they're making an immune response against Ebola virus. We don't know if that's enough to protect them against Ebola virus infection should they encounter the disease. So that's the very first stage of clinical development. This vaccine has to progress to be tested in the outbreak area, and ideally in the part of the world where cases were occurring, and it would be possible to see if this vaccine was stopping people getting infected with Ebola virus. So from our trials in Oxford, we passed on the information which facilitated the use of the vaccine in clinical trials in Mali, another West African country which was right next to the main outbreak area. And in this trial, healthcare workers were immunized, getting very, very similar data to that we generated in Oxford. Again, very positive results, ready to move into efficacy testing of the vaccine. And that was done by the end of 2014. So we had expected that this would then quickly go into the outbreak area, into a clinical trial to see if the vaccine worked. But what actually happened was that there was a lot of debate. The world was not ready to test a vaccine against a disease with such a high fatality rate. So it can be 70% mortality in those infected. That can be reduced somewhat by good medical support, and increasingly field hospitals were being set up and the patients were getting better and better care, but there are no specific drugs to treat Ebola. It's simply a case of providing support and keeping the patients quarantined so they can't infect anybody else. Unfortunately, no efficacy trials were initiated until April of 2015, and by that time the number of cases in the outbreak area were declining rapidly. Now that was great news for controlling the outbreak, but it wasn't great news for testing vaccines. The outbreak was controlled by just an intensive effort of contact tracing and quarantine. Cases dropped, and that meant that it was no longer possible to test more than one vaccine against Ebola. And as well as the one that we'd been working on, another one which had been brought up by Merck, a different viral vector vaccine, VSV vector vaccine, which is a different form of a virus, but expressing the same Ebola glycoprotein antigen. That was the one that went into efficacy testing first, with the few cases that were still remaining. Unfortunately, that was shown to have a very high efficacy. So the world then knew there was a vaccine that would work against Ebola, that would stop people getting infected. The other characteristics of that particular technology are not ideal for a vaccine to be used in West Africa. It requires ultra-low temperature storage, minus 80 degrees storage. The manufacturing process was not scaled up. It was still being manufactured in roller bottles. It's a very labor-intensive, small-scale process. It works to produce vaccines for clinical trials, but it's not a full-scale process that you would normally think of using for a licensed vaccine. So by the middle of 2015, the world had an Ebola vaccine that worked. It hadn't been possible to find out if the Chad3 vectored vaccine was effective or not, although looking at the immune responses, we would have expected that it would be. Subsequently, Johnson & Johnson licensed another Ebola vaccine based on comparison to the immune responses and using it to test in animals and protecting animals against deliberate Ebola virus infection. So there are now 2 licensed vaccines. The one that we had helped with was technology by that stage that was owned by GSK, and they decided not to pursue licensure for that particular Ebola virus vaccine. Another issue was that those vaccines only cover Ebola Zaire, and Ebola Sudan will also cause outbreaks in the same region of Africa, and these vaccines don't work, so we still didn't have a vaccine against Ebola Sudan. And we knew that eventually there would be another Ebola Sudan outbreak, and indeed last year there was. And again, we're in the same situation of having an outbreak in Africa and not having a vaccine. My colleague, Professor Theresa Lam, who's in the audience today, had been working on the early stages of development of a vaccine against Ebola Sudan, but hadn't had the funding to take that through to full-scale testing. It was then accelerated. The outbreak was then contained very quickly, and again, testing stopped. So we've been working in a very stop-start fashion. We know that there are viruses that regularly cause outbreaks in different parts of the world. These viruses normally are found in animals such as bats. Sometimes from bats they can transmit into a mammalian species, and then humans get exposed to the virus either directly from the bat or through the intermediate host, the mammalian species, and we can start to see outbreaks. But we don't have vaccines against them. And the small amount of vaccine development that had been done to this point didn't get us to the stage where we had a vaccine that we knew would be effective and we had a stockpile of the vaccine ready to use should there be an outbreak. And that's where we'd really like to be. So one of the other viruses that causes outbreaks is Nipah virus. Most of the cases are in Bangladesh, although it was first identified in Malaysia, carried in fruit bats. Fruit bats like to get into the pots that people use to collect date palm sap, and they can contaminate that with Nipah virus, and if you drink that date palm sap, you can become infected. 10 infections already since December in Bangladesh. That's this year's season. So it's 10 infections and counting. Very high fatality rate. It can also infect pigs. This is what happened in Malaysia when fruit bats were dropping contaminated fruit into the pig pens. The pigs got infected from that, and humans got infected through contact with the pigs. So we don't have a vaccine, and this disease has a very high fatality rate. We need to make a vaccine, and my group is now working on this using our own simian adenoviral vectored vaccine technology, ChAdOx1. The Nipah vaccine has now been manufactured, and we will be initiating clinical trials later this year. This work was delayed by the pandemic, but we now really need to accelerate because we need to have a vaccine against Nipah virus. At the moment, this virus is not very good at transmitting between people. It doesn't do it very well because most of the time it lives in bats. But as we've seen with the coronavirus, if we give viruses the opportunity to practice transmitting between people, what happens is those viruses mutate, and the ones that are more transmissible are the ones that are selected. And so every time we allow one of these viruses from bats or from other species to infect a human, we're giving it that chance to develop in something that becomes much more transmissible, much more likely to spread rapidly. And so we need to be controlling these infections and stopping outbreaks very, very quickly. And one of the ways to do that is to have a vaccine stockpile ready to use so that as soon as we start to see cases of these diseases, we can use the vaccine stockpile and stop the spread. Another virus that I'm working on a vaccine for is Lassa fever virus, again found in West Africa, this time carried in this small rodent, the multimammate mouse. It likes to get into people's houses, and it will then contaminate their food stocks with Lassa virus, and people then become infected. Most of the cases occur in children, and they tend to be quite mild fevers, but they can leave quite a large percentage of children who recover from this illness permanently deaf, presenting a different healthcare problem for the countries, particularly Nigeria, where this virus causes infections. Up to 200 million people are probably at risk of Lassa fever virus infection. And again, we have no vaccines. There's another virus which you would be more familiar with, Middle East respiratory syndrome, which has caused 800 deaths to date in 27 countries. Initially, we were seeing fairly large outbreaks in hospitals in the Kingdom of Saudi Arabia. That situation has been much improved. There was one very large outbreak in South Korea, which was the result of one person traveling from South Korea to the Middle East, becoming infected, and then going back to South Korea, and caused an outbreak which spread to 4 different hospitals. Camels are now the source of zoonotic infections. This virus was probably originally a bat virus. It managed to infect some camels, probably in Africa. Again, there's a lot of trade in camels. As you will be familiar with, between the Horn of Africa and the Middle East, some infected camels moved. It spreads very easily between camels, and it also spreads easily from camels to humans, because the receptor that the virus uses to get into cells is very, very similar in the camel and the human. So having made a rather slow adaptation from bats to camels, it's then able to make a rapid jump from camels to humans. And we know that occupational exposure to camels can result in seroconversion. People have antibodies in their bloodstream against MERS coronavirus without necessarily reporting any illness. And in young and healthy people, as with the SARS-2 coronavirus, it causes a mild disease. But if it gets into hospitals where there are many people who are immunocompromised, then MERS will cause severe and sometimes chronic infections with a high fatality rate, about a 30% fatality rate in the hospital outbreaks. The major surface antigen is the spike protein. Coronaviruses just have one protein on their surface, and so that's the protein that we were able to use to put into our ChAdOx1 adenoviral vector, a vector that is safe to use because it doesn't spread through the body after vaccination. It delivers the target antigen that we want an immune response against, and then it stops, and it can't spread through the body, and it can't transmit from person to person. So we'd been working on our MERS vaccine for some years. We'd conducted a clinical trial in the UK and shown that we could get antibodies and T cells, and including neutralizing antibodies, raised against the spike protein of the MERS coronavirus. We worked with staff at Kaimak to conduct a second clinical trial, and this was actually the first phase 1 vaccine trial that had ever been run in the Kingdom of Saudi Arabia, so we worked very closely with the Saudi FDA to receive approvals for that trial. That trial was also completed, and the data from that trial was also very positive, and that has also been published. So we were slowly moving through the process of developing the MERS coronavirus vaccine, working together with the spin-out company that I co-founded in Oxford called Vaccitech, which is going to assist in the further development of the MERS vaccine. But then in 2020, we had a different coronavirus to deal with. At the very end of 2019, a novel coronavirus started to cause cases of pneumonia of unknown cause in China. It was very quickly recognized that this was a viral infection, and it wasn't influenza. It wasn't adenovirus. It wasn't any of the viruses that we knew about. It was a coronavirus, and it wasn't a coronavirus that had ever caused cases in humans before. At the beginning of 2020, the genetic sequence of that coronavirus was released by Chinese scientists, and that's what vaccine developers around the world needed to initiate generation and testing of vaccines against this novel coronavirus. So with my team in Oxford, I worked very closely. We were waiting for the sequence to be released. We'd already decided that based on the successful use of the ChAdOx1 platform for other outbreak pathogens, in particular the MERS coronavirus vaccine, which being a coronavirus is very relevant to the SARS-CoV-2 outbreak as it was at the time, we decided that we would immediately start generating a vaccine to go into preclinical testing. We would plan GMP manufacture, so the very high quality manufacturing that we need to do to get into clinical trials. We would do the animal efficacy testing, also safety testing that we need to support clinical trials going forward. And we were able to start our clinical trials in April. Now the background that you can see on this slide, this grey triangle, is actually the exponential increase of the number of SARS-CoV-2 cases in the world as our development was going on. So when we started at the beginning of 2020, it was a case of, this looks like it might be interesting, but it might be contained very quickly. And then as we got into GMP manufacturing, it was, there are some more cases and it's spreading. And by the time we started our clinical trials in April of 2020, the UK was already in lockdown to try to prevent the further rapid spread of this coronavirus. And this gave us a problem because we hadn't ever considered how you do vaccine trials when you're in lockdown. So we had to do a lot of work to make sure that we could run these trials safely, as well as quickly introducing new measures like videos for participant information rather than face-to-face interviews, a lot of training, a lot of social distancing, bringing in mobile clinics so that we had more space to work in. But we were able to work through our phase 1 trial, and as with the MERS-CoV vaccine, show that we were getting neutralizing antibodies and T-cell response against the virus. We moved into phase 2 testing, expanding the age range of the people that we were vaccinating. And the next day it started phase 3 to look at the efficacy of the vaccine. And we worked with groups around the world to test the vaccine and find out if it was effective. And by November of 2020, we had the answer that it was. And by December of 2020, the MHRA had approved the vaccine for emergency use in the UK. So part of the work that we did in the trials involved getting information about the immunogenicity and also the efficacy of the vaccine with not just one dose, but with 2 doses, because we were able to show very quickly that the immune response was stronger when we gave 2 doses rather than 1. And we also showed that that increase was bigger if the interval between the 2 doses was not 1 month, but was 3 months. And so by the time the vaccine was licensed, we knew that the vaccine was effective. We knew that the first dose was effective, but the efficacy could be improved, and likely the duration of that efficacy could be maintained if we gave a second dose. Better to give it at 3 months than at 1 month. And this was also very important, taking into account the fact that although vaccine had been manufactured in readiness for rollout of the vaccine, this new process for large-scale manufacture takes some time to get up and running, and there was a shortage of all SARS-2 vaccines in January of 2021. So the best policy was to give as many people as possible, starting with the healthcare workers and those over the age of 80, their first dose, and then ask them to wait 3 months before they had their second dose. And by the 1st of March, so just over 2 years ago, it was possible to show that this policy was working extremely well. So this data was released at the beginning of March 2021, and looking at people over the age of 70 who just had their first dose of either the AstraZeneca vaccine, the Oxford AstraZeneca vaccine that we'd worked on, or the Pfizer vaccine, they were getting at least 60% protection against symptomatic PCR positive. So they have symptoms of coronavirus, they take a PCR test, yes, they are positive for coronavirus. That was being prevented in at least 60% of people over the age of 70 who'd had their first dose for either of those 2 vaccines. In the over 80s, hospitalization was reduced by 80%, again for either of those 2 vaccines, and the deaths in the over 80s were reducing faster than in the younger age groups which had not yet been vaccinated. So we've been able to develop a vaccine in a year. We've been able to get it rolled out and used in the population and showed that it protected not just young people, but the oldest members of society were very rapidly benefiting from being vaccinated. They went on to get their second dose at 3 months. But it wasn't just a vaccine for the UK. We had always intended this vaccine should be globally available, and AstraZeneca, who licensed the vaccine from us, worked very closely with us on the vaccine manufacturing scale-up, and they set up a global network of vaccine manufacturers and eventually had over 25 manufacturers in 15 countries. This meant that it was able to be sent around the world. It was the predominant vaccine used in the COVAX program for low and middle income countries. Since then, we formed the Pandemic Sciences Institute in Oxford because the vaccine wasn't the only thing that the Oxford University contributed in the pandemic. We also had the recovery trial led by Peter Horby, which showed which drugs were effective against hospitalized patients with SARS-CoV-2 and which were not, very quickly showing that dexamethasone, cheap and widely available, is effective. Hydroxychloroquine does not work. But we still have these other outbreak pathogens that we need to make vaccines for, and we don't want to go back to the old way of doing slow, stop-start process development, which takes up 10 years to get a vaccine licensed and ready for use. We want to think about what did we learn, how can we compress these timelines, how can we make sure that we can get these vaccines available now for the diseases that we already know about, have the efficacy testing done and the stockpiles ready, and how can we be best prepared to make new vaccines against the diseases that we don't yet know about. And the way we will approach that is through a project where we will work on having vaccines against examples of viruses from each of the known families, so that if the new virus that comes to form the next pandemic is not one we've seen before, it's highly likely it will be related to something that we've already got a vaccine for, and that will tell us the best approach to take to developing a new vaccine. So we hope to be much better prepared in future, and in the Pandemic Sciences Institute, we won't just work on vaccine development, but also diagnostics, therapeutics, clinical trials. Social scientists will also help us work on issues such as vaccine confidence. So we want to do all of the work that we need to be much better prepared for the next outbreak, preventing it from becoming a pandemic, or dealing with the next pandemic if we have to. And I'd like to finish by acknowledging the huge team in Oxford who came together from different groups very quickly and worked incredibly hard for many, many months, round the clock, in very difficult circumstances. We worked with some great clinical trial sites in Brazil and in South Africa that provided very high quality data that contributed to the vaccine licensure. All of the volunteers who took part in our clinical trials, without volunteers who are willing to come forward and receive vaccines, we will never know if these vaccines are safe or effective for widespread rollout. And of course the funders who provided the funding for us to do this work and make it globally available. By the beginning of 2022, this vaccine was estimated to save 6 million lives globally. 3 billion doses had been produced, and the production continues, and the number of lives saved will continue. We hope to be able to not get into that situation again, but if we ever have to deal with a new pandemic, we hope to be better prepared next time. Thank you.
Thank you. Are there any questions?
Those instances of microtrophs in some AstraZeneca recipients, can you say anything about those? Were those due to the vaccine itself?
So after the vaccine had been rolled out in many millions of doses, we did start to see very rare occurrences of thrombosis with thrombocytopenia in some vaccine recipients. More research has been done on that. We still don't have a mechanism for the exact cause of that. It's difficult to study because it is so very rare. It turns out that it only occurs after the first dose. We're back down to background rates. This is a phenomenon that is not unique to vaccination, but there was a higher rate after the first dose of the adeno-vectored vaccines. So anybody who's safely received their first dose will be fine to receive further doses of adeno-vectored vaccines. And we also see a global distribution with higher rates in Northern Europe and a gradation down to undetectable rates in the global south. So we're learning more about this phenomenon. Difficult to study because it's so rare, but certainly not an issue in some countries.
Are there any other questions? Thank you so much, Professor Sarah Gilbert. I would like to request the Advisor leadership to please come